this is basically the same way I went through it in class. I'm just going to go through it, and then so I'm going to go, what I'm going to do is I'm going to do light-dependent reaction, then I'm going to do Calvin's cycle, I'm going to come back, I'm going to erase this, so I don't have enough space to have it all up here, and then I'll do fermentation and cell respiration. Okay? So I'll do all the things that we've talked about. Alright, so if we start off with the light-dependent reactions, okay, again, just putting ourselves, making a setting for where we are in the thylakoid membrane, remember. We're in a chloroplast, in a cell, probably one of the palisade cells right in a leaf where there's quite a few chloroplasts. But we're in a chloroplast on a thylakoid membrane. The outside, stroma, the inside is the lumen. Okay. Now, is it helpful if I use those manipulatives? Or do you want me to just draw it?
Draw it and talk.
Okay, folks, here we have, I'm simplifying this, remember, a little bit. So here we have chlorophyll. Okay, so when we start out, we have an electron at low energy here. And I'm going to use black to indicate the electron. So here, we start out with chlorophyll. We have an electron that's at low energy. Now, initially we get, use orange for some photons here. From the sun, right, we get photons coming in. These photons are making their way in, one of which will strike our electron. The energy from the photon is transferred to the electron. The electron goes up in energy. So this electron goes up in energy. Now we have an electron that is high energy. So I'm going to write that here, high energy. Now that we have an electron that's high energy, we can actually do the electron transport chain, which is the idea that this molecule is going to go down in energy slowly, jumping from one molecule to another. Now, the molecules it jumps from and the molecules it jumps to are not important for this class. You don't need to know them. You just need to know that this electron is going to go through a series of steps where it's going to give off its energy. So, we typically like to draw the electron transport chain sort of like a series of stairs. Now, realize that this isn't true in terms of the spatial organization, right? The electron isn't actually going down the thylakoid membrane. This is representing the energy, right? It has high energy, and at the end it ends in low energy. Okay, so it's going along. All the while, that energy is being used, remember, to take hydrogen ions that are in the stroma, and it's being used for active transport. It's pumping them into the lumen this entire time. So the lumen is filled up with a much higher concentration of hydrogen than the outside. So the energy that was once in the photon, that was transferred to the electron, is now transferred into potential energy across this membrane. Right, we have potential energy because we have all these hydrogen ions here that want to go back. They want to go back across. Okay. Now again, we have another, so this electron has moved sort of down. Okay, so we have an electron that moves down. Another photon strikes it. So this could come from, pretty much, I'm not going to, you'll know the color coordinating. Another strikes it, it goes up in energy again. So we have E minus. And it goes up. It goes down another electron transport chain. Well, it's the same electron transport chain. Okay, it's just at the very end we have a molecule called NADP+. This is a final electron acceptor, right? It's going to take this electron Now remember, this electron jumps onto this NADP plus in the stroma. Okay? Just because it's down here in my drawing does not mean that it's where it would be realistically in this membrane. It would probably be up here and it would jump on. What happens is this electron jumps onto our NAD plus turns into NADPH. Now if I extend this a little bit.
The NAD plus is in the stroma, right?
Yes, this is in the stroma. This is all stroma. Everything above this line is stroma. Let's see. At this point, we've made one of our energy-carrying molecules, NADPH. However, this entire time, because we've been pumping hydrogen ions across, we've been letting them flow back across. I'm going to draw that down here. So these hydrogen ions, these protons can float back across, and all the while, that energy is being used to take ADP and turn it into ATP. So remember, the energy that was initially in this photon has now been transferred into ATP and NADPH. So all that's happening here is really it's a transfer of energy or a transforming of energy into a different form, into a chemical form that the plant can use in the Calvin cycle. So what happens is, let me draw that in a different color. NADPH and ATP go over here to our Calvin cycle.